Dear viewers, welcome to Massive Open Online Courses. In this segment, we will speak about riboviruses with reference to TMV, tobacco mosaic virus. First, riboviruses. Riboviruses are the nucleoproteinaceous entities that lie at borderline between non-living beings and living organisms and have RNA as genetic material. They have molecular level of organization and behave as non-living entities in extracellular environment, but show the features of living organisms in intracellular environment. They are non-cellular, ultramicroscopic, infectious particles. They have different types. On the basis of life cycle, they are of two types. One, retroviruses. Second, non-retroviruses. Retroviruses are those riboviruses which undergo reverse transcription inside host cell. They form DNA from RNA. The best example of retroviruses is HIV, human immunovirus that causes AIDS. Another group of riboviruses is the replicatoviruses or non-retroviruses. They are the viruses which, when inject their RNA into the host cell, allow the RNA molecule to replicate, but do not form DNA from the RNA. The best example is tobacco mosaic virus. Riboviruses can have single-stranded RNA, as is HIV, or as is TMV, or they can have double-stranded RNA. The best example of riboviruses is tobacco mosaic virus. It's a ribovirus which completes its life cycle in tobacco plants. This virus is a plant virus that infects tobacco and other plants and causes tobacco mosaic disease. It's a ribovirus with lytic type of life cycle. This means when it injects its RNA into the host cell, it bursts the cell after its replication. So far as this discovery is concerned, TMV was discovered by Ivanovsky in 1892. But its isolation from infected plants and its crystallization was done by W.M. Stanley in 1935. So far as the structure is concerned, each TMV particle appears as a rod, but in clusters it appears as a bundle of rods or needle-like structures. Each rod is approximately 300 nanometers long, 18 nanometers in diameter, and has a molecular weight of about 4 into 10 raised power 6 Daltons. Electron microscopy and X-ray crystallography has shown that the virus has two constituents, a protein coat and RNA core. The protein coat is called capsid and the core possesses single-stranded RNA. So far as capsid is concerned, it is made up of 2134 identical protein subunits called capsomers. Each capsomer consists of a long chain of 158 amino acids and has a molecular weight of 18,000 Daltons. The capsomers are helically arranged in a central stranded around RNA molecule with 16.3 capsomers per turn. So far as RNA core is concerned, it consists of single-stranded RNA molecule organized like a coil around a central cavity. It constitutes 5% of the total weight of the virion and possesses 6,401 nucleotides. The protein and nucleic acid ratio in these particles is 94.4 is to 5.6. In each helix of RNA, there are 49 nucleotides, and the helix has a pitch of 2.3 nanometers. The genetic information necessary for the formation of a complete TMV particle is contained in its RNA. This information determines the replication of RNA and the sequence of amino acids in the protein subunits of the capsid. As all the subunits are identical, only one coded gene is required for removing the protein coat from RNA. The naked RNA is capable of infecting tobacco plant once inside the host cell. The virus RNA directs the protein synthesizing apparatus of the host cell to synthesize its own proteins. Thus, RNA has two functions: self-replication and synthesis of specific virus proteins, for which it takes raw material from the host cell. Sources of infection. The virus particles are themselves the sources of infection. Infection to tobacco plants spreads through the debris of infected plants lying in the soil and the leaves fallen off from the plants. The infection also passes from one host to the other by mechanical means, such as agricultural implements and rubbing of infected leaves over the surface of healthy leaves. The virus is highly contagious and stable and can retain infectivity even after the leaves have been processed into smoking tobacco. According to Johansson, 1937, 67% of cigar, 11% cigarettes, and 62% pipe tobacco is virus infected. Symptoms of TMV on the host. Once the virus attacks the tobacco plants, especially leaves, what are the first symptoms? The first visible symptoms of TMV infection in the tobacco plant appear in the form of downward curling and distortion of young apical leaves. Large spots of dark green color appear on upper surface of leaves, which eventually develop into irregular blistered areas. Other areas of leaf show typical yellow and green mottling or mosaic symptoms. These are the pictures of the infected leaves and the virus attacking them. The infected leaves develop dark pads and start necrosis. After necrosis, they start dying and then fall off. Then the viruses are released. Once viruses are released, they spread to other plants and the infection cycle is repeated. Economic importance of viruses. Viruses are nucleoprotein entities that lie at borderline between living and non-living beings. They have been exploited on economical basis. A number of viruses have been extracted and have been used to produce vaccines. Basically, all viruses are superparasites and cause diseases in host cells. After understanding the diseases caused by them, biologists have isolated the viruses. They have prepared their weakened forms, which are used to stop the spread of diseases. Virus-based vaccine production is one of the major industrial endeavors related to medicine. Hepatitis B vaccine, oral polio vaccine, and a number of vaccines are available which help us in preventing spread of viral diseases. The other economic important roles played by viruses are they serve as biopesticides, they act as biomachines, they serve as protective agents, defensive agents, and play a role in phage typing. So far as prevention of industrial microbes from being attacked, we have a number of industrial microbes which are used as potential sources for the production of DNA, antibiotics, and the chemicals of importance in different fields. These microbes, if infected by viruses, lead to losses. To reduce the loss and to avoid infection, a number of viruses are being used in chemical industries to prevent the microbes from getting attacked. Second, during crop production, the plants are often infected in the field by either bacteria and other microbes, fungi, insects, and other organisms called pests. They reduce the crop production either by killing the plants or making them sick so that productivity decreases. In order to increase agricultural output and reduce the impact of disease-causing agents, a number of viruses have been identified, isolated, cultured, and made available for farmers so that their spray on the crop plants decreases the incidence of diseases and increases the agricultural output. A number of viruses serve as biomachines in genetic engineering. They are used as the agencies wherein DNA molecules are loaded, and these DNA molecules are carried to the target cells so that genetic modification is made. Transgenic plants and genetically modified organisms are produced with the help of insertion of foreign genes from the selected organisms with the help of viruses. Lysogenic viruses are a class of viruses which are used as biomachines. They are allowed to infect the cells where DNA is to be taken out. They are made able to cut the DNA segments, load them into their own DNA, and are used then to transfer the DNA segment into the selected organism, especially a plant. This process has helped agriculturists in increasing the agricultural output, in enhancing the quality, increasing the nutritious value, developing disease resistance, and tolerance against pests. Viruses have also been used as protective agents. They are added to paints, plasters, laminated wood to decrease the incidence of fungal and other pathogenic infections. High quality paints definitely contain viral agents which resist the impact of pests, especially the mites and fungal agencies. Thus, the durability of paints and wood has been increased. Viruses also serve as defensive agents. This earth is beamed with huge number of organisms, many of which are potential parasites and cause serious diseases in human beings, animals, and plants. Viruses have been identified which are specific to these parasitic organisms. They attack and destroy the cells of these parasitic organisms and therefore prevent the occurrence of diseases. Other role played by viruses is phage typing. Viruses have genetic material and they help in tracing out the genetic material of the unknown cells, unknown sites, unknown sources. They help in getting the knowledge of sequence of nucleotides in unknown fragments of DNA molecules. Other role played by viruses is identification of infectious agents. Many viruses have been used as markers to identify the presence of infectious agents in human body and the body of other animals. If a virus is injected into the body, it attacks a particular cell having a particular protein which provides it attachment site. The virus that attacks a specific cell and gets attached with the cell is used as a marker for tracing out presence of a pathogen in the body cells or in body tissues. Other use of viruses is that they help us in the production of virus-free plants. Viruses play a role as pathogens and cause diseases of plants. Tissue culture technique has been adopted and is being used to produce virus-free plants. First, the incidence of a disease caused by a virus is identified, and then the virus infected shoe tips are selected. They are placed on a nutrient medium, allowed to grow. As the shoe tips grow, the virus infected part is left behind towards the medium. The upper part is cut regularly until a stage is reached when the shoe tips become virus free. Once the shoe tips become virus free, they are then carried to the other nutrient media where they are allowed to produce roots, and the rooted plants are then sown in the soil as virus free plants. These are some of the uses of viruses of economic importance. Thank you.